Springtail. Springtails, class Collembola, form the largest of the three lineages of modern hexapods that are no longer considered insects. Although the three orders are sometimes grouped together in a class called Archeognaa because they have internal mouthparts, they do not appear to be any more closely related to one another than they are to all insects, which have external mouthparts. Springtails are omnivorous, free-living organisms that prefer moist conditions. They do not directly engage in the decomposition of organic matter, but contribute to it indirectly through the fragmentation of organic matter, and the control of soil microbial communities. 3. The word Collembola is from the ancient Greek kala, glue, and aimbolos, peg. This name was given due to the existence of the coliphore, which was previously thought to stick to surfaces to stabilize the creature. 4. Early DNA sequence studies 5, 6, 7 suggested that Collembola represent a separate evolutionary line from the other hexapoda, but others disagree. 8. This seems to be caused by widely divergent patterns of molecular evolution among the arthropods. 9. The adjustments of traditional taxonomic rank for springtails reflect the occasional incompatibility of traditional groupings with modern cladistics. When they were included with the insects, they were ranked as an order. As part of the Archeognaa, they are ranked as a subclass. If they are considered a basal lineage of hexapoda, they are elevated to full class status. Morphology. Isotoma angulicana. Entomobryomorpha, with visible furcula. Detonura monticola, peduromorpha. Members of the Collembola are normally less than 6 mm (0.24 in) long, have 6 or fewer abdominal segments, and possess a tubular appendage, the coliphore or ventral tube, with reversible, sticky vesicles, projecting ventrally from the first abdominal segment. 10. It is believed to be associated with fluid uptake and balance, excretion, and orientation of the organism itself. 11. Most species have an abdominal, tail-like appendage known as a furcula, or furca. It is located on the fourth abdominal segment of springtails and is folded beneath the body, held under tension by a small structure called the retinaculum, or tenaculum. When released, it snaps against the substrate, flinging the springtail into the air and allowing for rapid evasion and travel. All of this takes place in as little as 18 milliseconds. 12. 11. Springtails also possess the ability to reduce their body size by as much as 30% through subsequent ecdysis, molting, if temperatures rise high enough. The shrinkage is genetically controlled. Since warmer conditions increase metabolic rates and energy requirements in organisms, the reduction in body size is advantageous to their survival. 13. The peduromorpha and entomobryomorpha have an elongated body, while the Symphipleona and Nilipleona have a globular body. Collembola lack a tracheal respiration system, which forces them to respire through a porous cuticle, except for the two families Smithridae and Actyletidae, which exhibit a single pair of spiracles between the head and the thorax, leading to a springtails. Temporal range. Orchizella cincta. Scientific classification domain: Eukaryota kingdom, Animalia phylum, Arthropoda clade, Pancrustacea subphylum, Hexapoda class, Collembola. Lubbock, 1871. Orders: Entomobryomorpha, peduromorpha. Symphipleona. Nilipleona, disputed. Synonyms: 1. Oligentoma. Oligontoma rudimentary, although fully functional, tracheal system. 14.10. The anatomical variance present between different species partially depends on soil morphology and composition. Surface dwellers are generally larger, have darker pigments, have longer antennae and functioning furcula. Subsurface dwellers are usually unpigmented, have elongated bodies, and reduced furcula. They can be categorized into 4 main forms according to soil composition and depth: atmobiotic, epidaphic, hemidaphic, and eudaphic. Atmobiotic species inhabit macrophytes and litter surfaces. They are generally 8 to 10 mm, about 1/3, in length, pigmented, have long limbs, and a full set of ocelli photoreceptors. Epidaphic species inhabit upper litter layers and fallen logs. They are slightly smaller and have less pronounced pigments, as well as less developed limbs and ocelli than the atmobiotic species. Hemidaphic species inhabit the lower litter layers of decomposing organic material. They are 1 to 2 mm, about 1/16, in length, have dispersed pigmentation, shortened limbs, and a reduced number of ocelli. Eudaphic species inhabit upper mineral layers known as the humus horizon. They are smaller than hemidaphic species, have soft, elongated bodies, lack pigmentation and ocelli, and have reduced or absent furca. 15, 16, 17. Peduromorphs are characterized by their elongated bodies and conspicuous segmentation: 3 thoracic segments, 6 abdominal segments, including a well-developed prothorax with tergal chaetae, 17, while the first thoracic segment in Entomobryomorpha is clearly reduced and bears no chaetae. The digestive tract of springtails consists of 3 main components: the foregut, midgut, and hindgut. The midgut is surrounded by a network of muscles and lined with a monolayer of columnar or cuboidal cells. Its function is to mix and transport food from the lumen into the hindgut through contraction. Many species of syntrophic bacteria, archaea, and fungi are present in the lumen. These different digestive regions have varying pH to support specific enzymatic activities and microbial populations. The anterior portion of the midgut and hindgut is slightly acidic, with a pH of approximately 6.0, while the posterior midgut portion is slightly alkaline, with a pH of approximately 8.0. Between the midgut and hindgut is an alimentary canal called the pyloric region, which is a muscular sphincter. 11. Malpighian tubules are absent. 18. Alicma fusca, Symphipleona, on rotting wood. Traditionally, the springtails were divided into the orders Arthropleona, Symphipleona, and occasionally also Nilipleona. The Arthropleona were divided into two superfamilies, the Entomobryoidea and the Podaroidea. However, recent phylogenetic studies show Arthropleona is paraphyletic. 19-20-21 Thus, the Arthropleona are abolished in modern classifications, and their superfamilies are raised in rank accordingly, being now orders Entomobryomorpha and the Poduromorpha. Technically, the Arthropleona are thus a partial junior synonym of the Collembola. 22. The term, Neopleona, is essentially synonymous with Symphipleona plus Neelipleona. 23. The Neelipleona was originally seen as a particularly advanced lineage of Symphipleona, based on the shared global body shape, but the global body of the Neelipleona is realized in a completely different way than in Symphipleona. Subsequently, the Neelipleona were considered as being derived from the Entomobryomorpha. Analysis of 18S and 28S rRNA sequence data, though, suggests that they form the most ancient lineage of springtails, which would explain their peculiar apomorphies. 8. This phylogenetic relationship was also confirmed using a phylogeny based on mtDNA 20 and whole genome data. 21. The latest whole genome phylogeny supporting 4 orders of Collembola. 21. Neelipleona, Poduromorpha, Symphipleona, Entomobryomorpha. Systematics and evolution. Springtails are attested to since the early Devonian. 24. The fossil from 400 million years ago, Rhyniella precursor, is the oldest terrestrial arthropod, and was found in the famous Rhynie chert of Scotland. Given its morphology resembles extant species quite closely, the radiation of the hexapoda can be situated in the Silurian, 420 million years ago or more. 25. Additional research concerning the coprolites, fossilized feces, of ancient springtails allowed researchers to track their lineages back some 412 million years. 11. Fossil Collembola are rare. Instead, most are found in amber. 26. Even these are rare and many amber deposits carry few or no Collembola. The best deposits are from the early Eocene of Canada and Europe, 27. Miocene of Central America, 28. And the mid-Cretaceous of Burma and Canada. 29. They display some unexplained characteristics. First, all but one of the fossils from the Cretaceous belong to extinct genera, whereas none of the specimens from the Eocene or the Miocene are of extinct genera. Second, the species from Burma are more similar to the modern fauna of Canada than are the Canadian Cretaceous specimens. There are about 3,600 different species. 30. Ecology. Eating behavior. Specific feeding strategies and mechanisms are employed to match specific niches. 31. Herbivorous and detritivorous species fragment biological material present in soil and leaf litter, supporting decomposition and increasing the availability of nutrients for various species of microbes and fungi. 32. Carnivorous species maintain populations of small invertebrates such as nematodes, rotifers, and other collembolan species. 11. 15. Springtails commonly consume fungal hyphae and spores, but also have been found to consume plant material and pollen, animal remains, colloidal materials, minerals and bacteria. 33. Predators. Springtails are consumed by mesostigmatin mites in various families, including Ascidae, Lalapidae, Parasitidae, Rhodocaridae and Vagaidae. 34. Cave-dwelling springtails are a food source for spiders and harvestmen in the same environment, such as the endangered harvestman Texilaraceae. 35. To protect themselves, some species have evolved chemical defenses. 36. Distribution. Springtails are cryptozoa frequently found in leaf litter and other decaying material. 37. Where they are primarily detritivores and microbivores, and one of the main biological agents responsible for the control and the dissemination of soil microorganisms. 38. In a mature deciduous woodland in temperate climate, leaf litter and vegetation typically support 30 to 40 species of springtails, and in the tropics the number may be over 100. 39. Snow flea. A species of Smintherinae, Symphipleona. Smintherinae. In sheer numbers, they are reputed to be one of the most abundant of all macroscopic animals, with estimates of 100,000 individuals per square meter of ground, 40 essentially everywhere on Earth where soil and related habitats, moss cushions, fallen wood, grass tufts, ant and termite nests occur. 41. Only nematodes, crustaceans, and mites are likely to have global populations of similar magnitude, and each of those groups except mites is more inclusive. Though taxonomic rank cannot be used for absolute comparisons, it is notable that nematodes are a phylum and crustaceans a subphylum. Most springtails are small and difficult to see by casual observation, but one springtail, the so-called snow flea, Hypogastrorenivicola, is readily observed on warm winter days when it is active and its dark color contrasts sharply with a background of snow. 42. In addition, a few species routinely climb trees and form a dominant component of canopy fauna, where they may be collected by beating or insecticide fogging. 43. 44. These tend to be the larger, greater than 2 species, mainly in the genera Entomobrya and Orchizella, though the densities on a per square meter basis are typically 1 to 2 orders of magnitude lower than soil populations of the same species. In temperate regions, a few species, e.g. Anurophorus spp, Entomobrya albocincta, Xenila xavieri, Hypogastrura arborea, are almost exclusively arboreal. 41. In tropical regions a single square meter of canopy habitat can support many species of Collembola. 12. The main ecological factor driving the local distribution of species is the vertical stratification of the environment. In woodland a continuous change in species assemblages can be observed from tree canopies to ground vegetation then to plant litter down to deeper soil horizons. 41. This is a complex factor embracing both nutritional and physiological requirements, together with behavioral trends. 45. Dispersal limitation. 46. And probable species interactions. Some species have been shown to exhibit negative 47 or positive 45 gravitropism, which adds a behavioral dimension to this still poorly understood vertical segregation. Experiments with peat samples turned upside down showed two types of responses to disturbance of this vertical gradient, called stayers and movers. 48. Dysartemina sp. On leaf. As a group, springtails are highly sensitive to desiccation because of their tegumentary respiration. 49. Although some species with thin, permeable cuticles have been shown to resist severe drought by regulating the osmotic pressure of their body fluid. 50. The gregarious behavior of Collembola, mostly driven by the attractive power of pheromones excreted by adults. 51. Gives more chance to every juvenile or adult individual to find suitable, better protected places, where desiccation could be avoided and reproduction and survival rates, thereby fitness, could be kept at an optimum. 52. Sensitivity to drought varies from species to species. 53. And increases during ecdysis. 54. Given that springtails molt repeatedly during their entire life, an ancestral character in Hexapoda, they spend much time in concealed microsites where they can find protection against desiccation and predation during ecdysis, an advantage reinforced by synchronized molting. 55. The high humidity environment of many caves also favors springtails, and there are numerous cave-adapted species. 56, 57, including 1. Plutomyrus ortobologonensis living 1,980 meters, 6,500 feet down the Krubera cave. 58. Anorita maritima on water. The horizontal distribution of springtail species is affected by environmental factors which act at the landscape scale, such as soil acidity, moisture and light. 41. Requirements for pH can be reconstructed experimentally. 59. Altitudinal changes in species distribution can be at least partly explained by increased acidity at higher elevation. 60. Moisture requirements, among other ecological and behavioral factors, explain why some species cannot live above ground. 61. Or retreat in the soil during dry seasons. 62. But also why some epigale springtails are always found in the vicinity of ponds and lakes, such as the Hygrophilus Isotomyrus palustris. 63. Adaptive features, such as the presence of a fan-like wettable mucro, allow some species to move at the surface of water in freshwater and marine environments. 64. Podura aquatica, a unique representative of the family Poduridae, and one of the first springtails to have been described by Carl Linnaeus, spends its entire life at the surface of water, its wettable eggs dropping in water until the non-wettable first instar hatches then surfaces. 65. A few genera are capable of being submerged, and after molting young springtails lose their water-repellent properties and are able to survive submerged underwater. 66. In a variegated landscape, made of a patchwork of closed woodland and open meadows, cereal crops environments, most soil-dwelling species are not specialized and can be found everywhere, but most epigale and litter-dwelling species are attracted to a particular environment, either forested or not. 41. 67. As a consequence of dispersal limitation, land use change, when too rapid, may cause the local disappearance of slow-moving, specialist species. 68. A phenomenon the measure of which has been called colonization credit. 69. 70. Relationship with humans. Tomoceras sp. From Germany. Springtails are well known as pests of some agricultural crops. Sminthurus viridis, the lucerne flea, has been shown to cause severe damage to agricultural crops. 71. And is considered as a pest in Australia. 72. 73. Onychiridae are also known to feed on tubers and to damage them to some extent. 74. However, by their capacity to carry spores of mycorrhizal fungi and mycorrhiza helper bacteria on their tegument, soil springtails play a positive role in the establishment of plant-fungal symbiosis and thus are beneficial to agriculture. 75. They also contribute to controlling plant fungal diseases through their active consumption of mycelia and spores of damping off and pathogenic fungi. 76. 77. It has been suggested that they could be reared to be used for the control of pathogenic fungi in greenhouses and other indoor cultures. 78. 79. Various sources and publications have suggested that some springtails may parasitize humans, but this is entirely inconsistent with their biology, and no such phenomenon has ever been scientifically confirmed, though it has been documented that the scales or hairs from springtails can cause irritation when rubbed onto the skin. 80. They may sometimes be abundant indoors in damp places such as bathrooms and basements, and incidentally found on one's person. More often, claims of persistent human skin infection by springtails may indicate a neurological problem, such as delusional parasitosis, a psychological rather than entomological problem. Researchers themselves may be subject to psychological phenomena. For example, a publication in 2004 claiming that springtails had been found in skin samples was later determined to be a case of pareidolia. That is, no springtail specimens were actually recovered, but the researchers had digitally enhanced photos of sample debris to create images resembling small arthropod heads, which then were claimed to be springtail remnants. 80, 81, 82, 83, 84. However, Steve Hopkin reports one instance of an entomologist aspirating an Isotoma species and in the process accidentally inhaling some of their eggs, which hatched in his nasal cavity and made him quite ill until they were flushed out. 39. In 1952, China accused the United States military of spreading bacteria-laden insects and other objects during the Korean War by dropping them from P-51 fighters above rebel villages over North Korea. In all, the U.S. was accused of dropping ants, beetles, crickets, fleas, flies, grasshoppers, lice, springtails, and stoneflies as part of a biological warfare effort. The alleged associated diseases included anthrax, cholera, dysentery, foul septicemia, paratyphoid, plague, scrub typhus, smallpox, and typhoid. China created an international scientific commission for investigating possible bacterial warfare, eventually ruling that the United States probably did engage in limited biological warfare in Korea. The U.S. government denied all the allegations, and instead proposed that the United Nations send a formal inquiry committee to China and Korea, but China and Korea refused to cooperate. U.S. and Canadian entomologists further claimed that the accusations were ridiculous and argued that anomalous appearances of insects could be explained through natural phenomena. 85 springtail species cited in allegations of biological warfare in the Korean War were Isotoma, Desoria, Negashina, a local species, and the white rat springtail, Folsomia candida. 86 captive springtails are often kept in a terrarium as part of a cleanup crew. 87 ecotoxicology laboratory animals. Springtails are currently used in laboratory tests for the early detection of soil pollution. Acute and chronic toxicity tests have been performed by researchers, mostly using the parthenogenetic isotomid Folsomia candida. 88 These tests have been standardized. 89 Details on a ring test, on the biology and ecotoxicology of Folsomia candida and comparison with the sexual nearby species Folsomia Ephemeroptera, sometimes preferred to Folsomia candida, are given in a document written by Paul Henning Krogh. 90 Care should be taken that different strains of the same species may be conducive to different results. Avoidance tests have been also performed. 91 They have been standardized, too. 92 Avoidance tests are complementary to toxicity tests, but they also offer several advantages. They are more rapid, thus cheaper, more sensitive, and they are environmentally more reliable, because in the real world Collembola move actively far from pollution spots. 93 It may be hypothesized that the soil could become locally depopulated in animals, and thus improper to normal use, while below thresholds of toxicity. Contrary to earthworms, and like many insects and mollusks, Collembola are very sensitive to herbicides and thus are threatened in no-tillage agriculture, which makes a more intense use of herbicides than conventional agriculture. 94 The springtail Folsomia candida is also becoming a genomic model organism for soil toxicology. 95-96 With microarray technology the expression of thousands of genes can be measured in parallel. The arthropods portal. Gene expression profiles of Folsomia candida exposed to environmental toxicants allow fast and sensitive detection of pollution, and additionally clarifies molecular mechanisms causing toxicology. Collembola have been found to be useful as bioindicators of soil quality. Laboratory studies have been conducted that validated that the jumping ability of springtails can be used to evaluate the soil quality of Quang Ni polluted sites. 97. In polar regions that are expected to experience among the most rapid impact from climate warming, springtails have shown contrasting responses to warming in experimental warming studies. 98 There are negative, 99, 100 positive, 101, 102, and neutral responses reported. 100, 103 neutral responses to experimental warming have also been reported in studies of nonpolar regions. 104 The importance of soil moisture has been demonstrated in experiments using infrared heating in an alpine meadow, which had a negative effect on mesofauna biomass and diversity in drier parts and a positive effect in moist sub-areas. 105 Furthermore, a study with 20 years of experimental warming in 3 contrasting plant communities found that small-scale heterogeneity may buffer springtails to potential climate warming. 103 Sexual reproduction occurs through the clustered or scattered deposition of spermatophores by male adults. Stimulation of spermatophore deposition by female pheromones has been demonstrated in Sinella crivaceta. 106 Mating behavior can be observed in Symphipleona. 107 Among Symphipleona, males of some Smithridae use a clasping organ located on their antenna. 37 Many springtails, mostly those living in deeper soil horizons, are parthenogenetic, which favors reproduction to the detriment of genetic diversity and thereby to population tolerance of environmental hazards. 108 Parthenogenesis, also called thalidoche, is under the control of symbiotic bacteria of the genus Wolbachia, which live, reproduce and are carried in female reproductive organs and eggs of Collembola. 109 Feminizing Wolbachia species are widespread in arthropods, 110, and nematodes, 111, where they co-evolved with most of their lineages. Sexual 103 Sexual reproduction occurs through the clustered or scattered deposition of spermatophores by male adults. Stimulation of spermatophore deposition by female pheromones has been demonstrated in Sinella crivaceta. 106 Mating behavior can be observed in Symphipleona. 107 Among Symphipleona, males of some Smithridae use a clasping organ located on their antenna. 37 Many springtails, mostly those living in deeper soil horizons, are parthenogenetic, which favors reproduction to the detriment of genetic diversity and thereby to population tolerance of environmental hazards. 108 Parthenogenesis, also called thalidoche, is under the control of symbiotic bacteria of the genus Wolbachia, which live, reproduce and are carried in female reproductive organs and eggs of Collembola. 109 Feminizing Wolbachia species are widespread in arthropods, 110, and nematodes, 111, where they co-evolved with most of their lineages. Protura. The Protura, or proturans, and sometimes nicknamed coneheads, 2-3, are very small, 0.6-1.5 mm long, soil-dwelling animals, so inconspicuous they were not noticed until the 20th century. The Protura constitute an order of hexapods that were previously regarded as insects, and sometimes treated as a class in their own right. 1, 4, 5. Some evidence indicates the Protura are basal to all other hexapods. 6. Although not all researchers consider them hexapoda, rendering the monophyly of hexapoda unsettled. 7. Uniquely among hexapods, proturans show anamorphic development, whereby body segments are added during molts. 8. There are close to 800 species, described in 7 families. Nearly 300 species are contained in a single genus, Eosintomen. 1. 9. Morphology. Proturans have no eyes, wings, or antennae, and, lacking pigmentation, are usually whitish or pale brown. The sensory function of the absent antennae is fulfilled by the first pair of the 3 pairs of 5 segmented legs, which are held up, pointing forward, and bearing numerous tarsal cincilla and sensory hairs. They ambulate using the 4 rear legs. 10. The head is conical, and bears 2 pseudoculi with unknown function. The body is elongated and cylindrical. 11. With a post-anal telson at the end. The mouthparts are entignathous, enclosed within the head capsule, and consist of narrow mandibles and maxillae. 8. There are no cerci at the end of the abdomen, which gives the group their name, from the Greek proto meaning first, in this case implying primitive, and ura, meaning tail. 12. The first 3 abdominal segments bear short limb-like appendages, 10 called styli. 12. The first pair of styli is 2-segmented, while the second and third pair are either 2-segmented or unsegmented. 5. The genitalia are internal and the genital opening lies between the 11th segment and the telson of the adult. 10. During mating, the genitalia of both sexes are everted from an abdominal chamber. 13. Only the 2 families Eosintomidae and Synentomidae possess a simple tracheal system with a pair of spiracles on both the mesothorax and the metathorax, while proturans in the remaining families lack these structures and perform gas exchange by diffusion. 10, 14, 15. Proturan found in Durham, United States. Proturans live chiefly in soil, mosses, and leaf litter. 8. Of moist temperate forests. 12. That are not too acidic. 16. They have also been found beneath rocks or under the bark of trees. 11. As well as in animal burrows. 10. They are generally restricted to the uppermost 0.1 meters, 3.9 in. 16. But have been found as deep as 0.25 meters, 9.8 in. 17. Although they are sometimes regarded as uncommon. 12. Proturans are most likely overlooked because of their small size. 11. As densities of over 90,000 individuals per square meter have been recorded. 18. The diet of proturans has not yet been sufficiently observed to be characterized. In laboratory culture, they may be fed mycorrhizal fungi, dead mites and pulverized, dried mushrooms. 10. They are believed to feed on decaying vegetable matter and fungi in the wild. 11. 12. The styliform mouthparts suggest the Protura may be fluid feeders, based on evidence that some species suck out the liquid contents of fungal hyphae. 16. Proturan species which spend their lives near the soil surface generally produce one new generation of offspring each year. They also possess longer legs. Species living at deeper soil levels have shorter legs and tend to reproduce less seasonally. Some Protura. Aceratomen sp. Under stereo microscope. Scientific classification domain. Eukaryota kingdom. Animalia phylum. Arthropoda clade. Pancrustacea subphylum. Hexapoda order. Protura. Silvestri, 1907. Families. 1. Aceratomatta. Hesperantomidae protentomidae Aceratomidae Eosintomatta. Anteliantomidae Eosintomidae Synentomatta. Fujiantomidae Synentomidae. Ecology. Migratory proturan species move to deeper soil layers for the winter and ascend to shallower soil layers for the summer. 16. Proturans play a role in soil formation and composition by speeding decomposition, helping in the breakdown of leaf litter and recycling nutrients into the soil. 19. Development. The nymph has 8 abdominal segments plus the telson. The number of abdominal segments increases through molting until the full adult complement of 11 abdominal segments is achieved. Further molts may occur, but do not add additional body segments. 12. It is still not known whether the adults continue to molt throughout their lives. 10. Eggs have been observed in only a few species. 10. In most proturan families, 5 developmental stages follow the egg stage. The prenymph hatches from the egg and has only weakly developed mouthparts and 8 abdominal segments. Nymph I follows and has fully developed mouthparts. Nymph 2 has 9 abdominal segments. Maturus junior has 11 abdominal segments and molts into the sexually mature adult. 10. Male individuals of the family Aceratomidae differ from this 5-stage scheme, having an additional developmental stage, the preimago, which has partially developed genitalia and appears between the maturus junior and the adult stage. 10. History. Proturans were first discovered in the early 20th century, when Filippo Silvestri and Antonio Berlese independently described the animals. 16. The first species to be described was Aceratomen dodoroi, published in 1907 by Silvestri. 10. Based on material found near Syracuse, New York. 12. The order Diplura, 2-pronged bristletails, is one of 3 orders of non-insect hexapods within the class Archeognaa, alongside Collembola, springtails, and Protura. 3. The name, Diplura, or 2 tails, refers to the characteristic pair of caudal appendages or filaments at the terminal end of the body. Around 800 species of diplurans have been described. Anatomy. A dipluran of the family Campodidae. Diplurans are typically 2 to 50 mm, 0.08-1.97 in long, with most falling between 7 and 10 mm, 0.28 and 0.39 in. 4. However, some species of Geopix may reach 50 mm, 2.0 in. 5. They have no eyes and, apart from the darkened cerci in some species, they are unpigmented. 5. Diplurans have long antennae with 10 or more bead-like segments projecting forward from the head. 6. The abdomens of diplurans bear eversible vesicles, which seem to absorb moisture from the environment and help with the animal's water balance. 6. The body segments themselves may display several types of setae, or scales and setae. 7. Diplurans possess a characteristic pair of cerci projecting backwards from the last of the 11 abdominal somites. 8. These cerci may be long and filamentous or short and pincer-like. 9. Leading to occasional confusion with earwigs. 10. Some diplurans have the ability to shed their cerci if necessary. Autotomy. Molting occurs up to 30 times throughout the life of a dipluran, which is estimated to last up to 1 year. As entignaths, the mouth parts are concealed within a small pouch by the lateral margins of the head capsule. In males, Diplura. The order Diplura, two-pronged bristletails, is one of 3 orders of non-insect hexapods within the class Archeognaa, alongside Collembola, springtails, and Protura. 3. The name, Diplura, or two tails, refers to the characteristic pair of caudal appendages or filaments at the terminal end of the body. Around 800 species of diplurans have been described. Anatomy. A dipluran of the family Campodidae. Diplurans are typically 2 to 50 mm, 0.081.97 in long, with most falling between 7 and 10 mm, 0.28 and 0.39. 4. However, some species of Gepics may reach 50 mm, 2.0 in. 5. They have no eyes and, apart from the darkened cerci in some species, they are unpigmented. 5. Diplurans have long antennae with 10 or more bead-like segments projecting forward from the head. 6. The abdomens of diplurans bear eversible vesicles, which seem to absorb moisture from the environment and help with the animal's water balance. 6. The body segments themselves may display several types of setae, or scales and setae. 7. Diplurans possess a characteristic pair of cerci projecting backwards from the last of the 11 abdominal somites. 8. These cerci may be long and filamentous or short and pincer-like. 9. Leading to occasional confusion with earwigs. 10. Some diplurans have the ability to shed their cerci if necessary. Autotomy. Molting occurs up to 30 times throughout the life of a dipluran, which is estimated to last up to 1 year. As entignaths, the mouth parts are concealed within a small pouch by the lateral margins of the head capsule. The mandibles usually have several apical teeth. 7. Diplurans do not possess any eyes or wings. 4. In males, glandular setae or disculi may be visible along the first abdominal sternite. External genital organs are present on the 8th abdominal segment. 7. Diplurans are common in moist soil, leaf litter or humus. 11. But are rarely seen because of their size and subterranean lifestyles. 6. They have biting mouthparts and feed on a variety of live prey and dead organic matter. 3. Those species with long cerci are herbivorous. 10. Diplurans are found on nearly all land masses, except Antarctica and several oceanic islands. 7. Their role as soil-dwelling organisms may play a key role in indicating soil quality, and as a measure of anthropogenic impact, e.g. soil nutrient depletion as a result of farming. 6. They have biting mouthparts and feed on a variety of live prey and dead organic matter. 3. Those species with long cerci are herbivorous. 10. Diplurans are found on nearly all land masses, except Antarctica and several oceanic islands. 7. Their role as soil-dwelling organisms may play a key role in indicating soil quality, and as a measure of anthropogenic impact, e.g. soil nutrient depletion as a result of farming. 12. 13. Like other non-insect hexapods, diplurans practice external fertilization. Males lay up to 200 spermatophores a week, which are held off the ground by short stalks and probably only remain viable for about 2 days. 11. The female collects the spermatophore with her genital opening, and later lays eggs in a cavity in the ground. 10. 6. 11. The hatchlings, or nymphs, do not undergo metamorphosis, but resemble the adults, apart from their smaller size, lesser number of setae and their lack of reproductive organs. 3. Diplurans. Temporal range. Campodia staphylinus, Belgium. Scientific classification domain, Eukaryota kingdom, Animalia phylum, Arthropoda clade, Pancrustacea subphylum, Hexapoda order, Diplura. Borner, 1904. Families, 2. Campodidae. Procampodidae Progepigidae. Anagepigidae Gepigidae. Heterohepigidae Dingepigidae Evelgepigidae Paragepigidae. Ecology. Reproduction. Anatomy of Campodia Campodidae and Gepics Gepigidae. Several major lineages within Diplura are readily recognizable by the structure of their cerci. Gepigidae possess forceps-like cerci, resembling those of an earwig. Usually very aggressive predatory diplurans, using their pincer-like cerci to capture prey, including springtails, isopods, small myriapods, insect larvae, and even other diplurans. 3. Progepigidae possess stout, short, and rigid cerci. 14. Campodidae possess elongate, flexible cerci that may be as long as the antennae and have many segments. Feed on soil fungi, mites, springtails, and other small soil invertebrates, as well as detritus. 3. The relationships among the 4 groups of hexapods are not resolved, but most recent studies argue against a monophyletic Archeognaa. 15. The fossil record of the Diplura is sparse, but one apparent dipluran dates from the Carboniferous. 2. This early dipluran, Testogepics, had compound eyes, and mouthparts that more closely resemble those of true insects. Archeognaa. The Archeognaa are an order of apterogoats, known by various common names such as jumping bristletails. Among extant insect taxa they are some of the most evolutionarily primitive. They appeared in the Middle Devonian period at about the same time as the arachnids. Specimens that closely resemble extant species have been found as both body and trace fossils, the latter including body imprints and trackways, in strata from the remainder of the Paleozoic era and more recent periods. 3. For historical reasons, an alternative name for the order is Microcoryphia. 4. Until the late 20th century, the suborders Zygentoma and Archeognaa comprise the order Thyasanura. Both orders possess three-pronged tails comprising two lateral cerci and a medial epiproctor appendix dorsalis. Of the three organs, the appendix dorsalis is considerably longer than the two cerci. In this, the Archeognaa differ from the Zygentoma, in which the three organs are subequal in length. 4. In the late 20th century, it was recognized that the order Thyasanura was paraphyletic, thus the two suborders were each raised to the status of an independent monophyletic order, with Archeognaa sister taxon to the Dicondylia, including the Zygentoma. 5. The order Archeognaa is cosmopolitan. It includes roughly 500 species in two families. 6. No species is currently evaluated as being at conservation risk. 7. Description. Archeognaa are small insects with elongated bodies and backs that are arched, especially over the thorax. Their abdomen ends in three long tail-like structures, of which the lateral two are cerci, while the medial filament, which is longest, is an epiproct. The 10th abdominal segment is reduced. 8. The antennae are flexible. The two large compound eyes meet at the top of the head, and there are three ocelli. The mouthparts are partly retractable, with simple chewing mandibles and 7 segmented maxillary palps which are commonly longer than the legs. 9. 341-343. Unlike other insect orders, they do not have olfactory receptor coreceptors, ARCO, which have either been lost or were never present in the first place. 10. 11. Archeognaa differ from Zygentoma in various ways, such as their relatively small head, their bodies being compressed laterally, from side to side, instead of flattened dorsiventrally, and in their being able to use their tails to spring up to 30 centimeters 12 in into the air if disturbed. They have 8 pairs of short appendages called styli on abdominal segments 2 to 9. Family Motullidae is also unique among insects in possessing small muscleless styli on the second and third thoracic legs, 12. But are absent on the second pair of thoracic legs in some genera. 13. Similar stylets on the legs are absent in family Minortellidae. 14. They have 1 or 2 pairs of eversible membranous vesicles on the underside of abdominal segments 1 to 7, which are used to absorb water and assisting with molting. 15. There are 9 pairs of spiracles, 2 pairs on the thorax, and 7 pairs on abdominal segments 2 to 8. 16. The pair of spiracles on the first abdominal segment has been lost. 17. Lateral aspect of an Archeognathan, showing arched profile and abdominal styli. Head of a motulid, showing compound eyes, prominent maxillary palps, and detachable scales. Archeognatha. Temporal range. Rock bristletail. Scientific classification domain, Eukaryota kingdom, Animalia phylum, Arthropoda class, Insecta subclass, Monocondylia. Haeckel, 1866. Order, Archeognatha. Borner, 1904. Families, 2. Monora, Sheriff 1957 Stat. November. Carpenter 1992. Dasyleptidae. Motulida, Grassi 1888 Stat. November. Zhang et al. 2018. Motullidae. Minortellidae. Synonyms. Microcoryphia, Verhoff 1904. Drawing of a scale, much magnified, from a species in the family Motullidae. Further unusual features are that the abdominal sternites are each composed of 3 sclerites, and they cement themselves to the substrate before molting, often using their own feces as glue. 18. The body is covered with readily detached scales, that make the animals difficult to grip and also may protect the exoskeleton from abrasion. The thin exoskeleton offers little protection against dehydration, and they accordingly must remain in moist air, such as in cool, damp situations under stones or bark. The name Archeognatha is derived from the Greek archaios, meaning ancient, and gnathos, meaning jaw. This refers to the articulation of the mandibles, which are different from those of other insects. It was originally believed that Archeognatha possessed a single phylogenetically primitive condyle each, thus the name, Monocondylia, where all more derived. Trinomora, Crampton 1916. Protothysanura, Crampton 1924, Non-Smith 1897. Etymology. Insects have 2, but this has since been shown to be incorrect. All insects, including Archeognatha, have dicondylic mandibles, but Archeognats possess 2 articulations that are homologous to those in other insects, though slightly different. 19. An alternative name, Microcoryphia, 4 comes from the Greek mikros, small, and koryphi, which in context means head. 20. Taxonomy. Families number of species defining notes image Motullidae 250 most are restricted to rocky shorelines need specific image Minortellidae 170, 21 lack scales at base of hind legs and antennae need specific image. Biology. Archeognatha occur in a wide range of habitats. While most species live in moist soil, others have adapted to chaparral, and even sandy deserts. They feed primarily on algae, but also lichens, mosses, or decaying organic detritus. 3 types of mating behavior are known. In some species the male spins a silk thread, carrier thread, stretched out on the ground. On the thread there are droplets of sperm which the female will take up when her ovipositor makes contact. In others a packet of sperm, spermatophore, is deposited on the top of a short stalk. If a female takes up the sperm or not is often random, but in many species the male will try to lead the female's genitals over the sperm during courtship. A more direct way of fertilization occurs in species of the genus Petrobius, where the male places a droplet of sperm directly on the female's ovipositor. 22-23 One hypothesis is that the external genitals of insects started as structures specialized for water uptake, which could reach deeper crevices than the coxal vesicles, and over time the female would use it to take up sperm from the ground instead of water. 24 After fertilization she lays a batch of around 30 eggs in a suitable crevice. The young resemble the adults, and take up to 2 years to reach sexual maturity, depending on the species and conditions such as temperature and available food. Unlike most insects, the adults continue to molt after reaching adulthood, and typically mate once at each instar. Archeognats may have a total lifespan of up to 4 years, longer than most larger insects. 1. Archeognatha. The Archeognatha are an order of apterogoats, known by various common names such as jumping bristletails. Among extant insect taxa they are some of the most evolutionarily primitive. They appeared in the Middle Devonian period at about the same time as the arachnids. Specimens that closely resemble extant species have been found as both body and trace fossils, the latter including body imprints and trackways, in strata from the remainder of the Paleozoic era and more recent periods. 3. For historical reasons an alternative name for the order is Microcoryphia. 4. Until the late 20th century the suborders Zygentoma and Archeognatha comprise the order Thyasanura. Both orders possess 3-pronged tails comprising 2 lateral cerci and a medial epiprocta or appendix dorsalis. Of the 3 organs, the appendix dorsalis is considerably longer than the 2 cerci. In this the Archeognatha differ from the Zygentoma, in which the 3 organs are subequal in length. 4. In the late 20th century, it was recognized that the order Thyasanura was paraphyletic, thus the 2 suborders were each raised to the status of an independent monophyletic order, with Archeognatha sister taxon to the Dicondylia, including the Zygentoma. 5. The order Archeognatha is cosmopolitan. It includes roughly 500 species in 2 families. 6. No species is currently evaluated as being at conservation risk. 7. Description. Archeognatha are small insects with elongated bodies and backs that are arched, especially over the thorax. Their abdomen ends in 3 long tail-like structures, of which the lateral 2 are cerci, while the medial filament, which is longest, is an epiproct. The 10th abdominal segment is reduced. 8. The antennae are flexible. The 2 large compound eyes meet at the top of the head, and there are 3 ocelli. The mouthparts are partly retractable, with simple chewing mandibles and 7 segmented maxillary palps which are commonly longer than the legs. 9. 341-343. Unlike other insect orders, they do not have olfactory receptor coreceptors, ARCO, which have either been lost or were never present in the first place. 10. 11. Archeognatha differ from Zygentoma in various ways, such as their relatively small head, their bodies being compressed laterally, from side to side, instead of flattened dorsiventrally, and in their being able to use their tails to spring up to 30 cm into the air if disturbed. It's called styli on abdominal segments 2 to 9. Family Mochilidae is also unique among insects in possessing small muscleless styli on the 2nd and 3rd thoracic legs, 12. But are absent on the 2nd pair of thoracic legs in some genera. 13. Similar stylets on the legs are absent in family Minortelidae. 14. They have 1 or 2 pairs of eversible membranous vesicles on the underside of abdominal segments 1 to 7, which are used to absorb water and assisting with molting. 15. There are 9 pairs of spiracles, 2 pairs on the thorax, and 7 pairs on abdominal segments 2 to 8. Thyasanura is the now deprecated name of what was, for over a century, recognized as an order in the class Insecta. The 2 constituent groups within the former order, the Archeognatha jumping bristletails and the Zygentoma silverfish and firebrats, share several characteristics, such as of having 3 long caudal filaments, the lateral ones being the cerci, while the one between telson is a medial cerciform appendage, specifically an epiproct. They are also both wingless, and have bodies covered with fine scales, rather like the scales of the practically unrelated Lepidoptera. In the late 20th century, it was recognized that the 2 suborders were not sister taxa, therefore Thyasanura was paraphyletic, and the 2 suborders were each raised to the status of an independent monophyletic order, with Archeognatha sister taxon to the Dicondylia, including the Zygentoma. 1. 2. A typical member of the order Archeognatha. Although the group Thyasanura is no longer recognized, the name still appears in some published material. 3. Another name used to separate the 2 groups from winged insects is Apterogota. Etymology. The name Thyasanura, first applied to the group by Pierre-André Latreille, 4, was derived from the Greek thasanos for fringe, tassel, bristle and aura for, tail, a reference to the 3 fanned-out caudal filaments. This etymology is consistent with the English word, bristletail, which is the common name for several hexapod species, not all of which fell within Thyasanura. Mayflies, also known as shadflies or fishflies in Canada and the upper Midwestern United States, as Canadian soldiers in the American Great Lakes region, 2, and as upwing flies in the United Kingdom, are aquatic insects belonging to the order Ephemeroptera. This order is part of an ancient group of insects termed the Paleoptera, which also contains dragonflies and damselflies. Over 3,000 species of mayfly are known worldwide, grouped into over 400 genera in 42 families. Mayflies have ancestral traits that were probably present in the first flying insects, such as long tails and wings that do not fold flat over the abdomen. Their immature stages are aquatic freshwater forms called naiads, or nymphs, whose presence indicates a clean, unpolluted and highly oxygenated aquatic environment. They are unique among insect orders in having a fully winged terrestrial pre-adult stage, the subimago, which molts into a sexually mature adult, the imago. Mayflies hatch, emerge as adults from spring to autumn, not necessarily in May, in enormous numbers. Some hatches attract tourists. Fly fishermen make use of mayfly hatches by choosing artificial fishing flies that resemble them. One of the most famous English mayflies is Archeognaa germanica, the fisherman's March brown mayfly. 3. The brief lives of mayfly adults have been noted by naturalists and encyclopedists since Aristotle and Pliny the Elder in classical antiquity. The German engraver Albrecht Dürer included a mayfly in his 1495 engraving the Holy Family with the mayfly to suggest a link between heaven and earth. The English poet George Crabbe compared the brief life of a daily newspaper with that of a mayfly in the satirical poem, The Newspaper, 1785, both being known as ephemera. Description. Nymph. Top left, mayfly nymph, dorsal view, showing the paired gills and 3 projections on the abdomen, wing buds are visible on the thorax. Top right, subimago of Leptophlebia marginata. Bottom, nymph of the mayfly Collembola, showing 7 pairs of gills along the sides of the abdomen. Bottom, nymph of. Top right, subimago of Leptophlebia marginata. Bottom. Top left, mayfly nymph, dorsal view, showing the paired gills and 3 projections on the abdomen, wing buds are visible on the thorax. Top right, subimago of Leptophlebia marginata. Bottom, nymph of the mayfly Collembola, showing 7 pairs of gills along the sides of the abdomen. Immature mayflies are aquatic and are referred to as nymphs or naiads. In contrast to their short lives as adults, they may live for several years in the water. They have an elongated, cylindrical or somewhat flattened body that passes through a number of instars, stages, molting and increasing in size each time. When ready to emerge from the water, nymphs vary in length, depending on species, from 3 to 30 mm, 0.12 to 1.18 in. 4. The head has a tough outer covering of sclerotin, often with various hard ridges and projections. It points either forwards or downwards, with the mouth at the front. There are 2 large compound eyes, 3 ocelli, simple eyes, and a pair of antennae of variable lengths, set between or in front of the eyes. The mouth parts are designed for chewing and consist mayfly. Temporal range. Archeognaa germanica, the fly fisherman's March brown mayfly. Scientific classification domain, Eukaryota kingdom, Animalia phylum, Arthropoda class, Insecta subclass, Pterygoda division, Coleoptera superorder, Ephemeropteroidea. Rodendorf, 1968. Order, Ephemeroptera. Hyatt and Arms, 1890. Suborders and families. See text. Of a flap-like labrum, a pair of strong mandibles, a pair of maxillae, a membranous hypopharynx and a labium. 5. The thorax consists of 3 segments, the hindmost 2, the mesothorax and metathorax, being fused. Each segment bears a pair of legs which usually terminate in a single claw. The legs are robust and often clad in bristles, hairs or spines. Wing pads develop on the mesothorax, and in some species, hindwing pads develop on the metathorax. 5. The abdomen consists of 10 segments, some of which may be obscured by a large pair of operculate gills, a thoracic shield, expanded part of the prothorax, or the developing wing pads. In most taxa up to 7 pairs of gills arise from the top or sides of the abdomen, but in some species they are under the abdomen, and in a very few species the gills are instead located on the coxae of the legs, or the bases of the maxillae. The abdomen terminates in slender thread-like projections, consisting of a pair of cerci, with or without a third central caudal filament. 5. Subimago. The final molt of the nymph is not to the full adult form, but to a winged stage called a subimago that physically resembles the adult, but which is usually sexually immature and duller in color. The subimago, or dun, 6, often has partially cloudy wings fringed with minute hairs known as microtrichia. Its eyes, legs and genitalia are not fully developed. Females of some mayflies, subfamily Palingoninae, do not molt from a subimago state into an adult stage and are sexually mature while appearing like a subimago with microtrichia on the wing membrane. Oligonorine mayflies form another exception in retaining microtrichia on their wings but not on their bodies. Subimagos are generally poor flyers, have shorter appendages, and typically lack the color patterns used to attract mates. In males of Ephor and Lucan, the subimagos have forelegs that are short and compressed, with accordion-like folds, and expands to more than double its length after molting. 7. After a period, usually lasting 1 or 2 days but in some species only a few minutes, the subimago molts to the full adult form, making mayflies the only insects where a winged form undergoes a further molt. 4. Imago. Adult Ataliphlebia with the cylindrical dorsal or turban eyes visible. Adult mayflies, or imagos, are relatively primitive in structure, exhibiting traits that were probably present in the first flying insects. These include long tails and wings that do not fold flat over the abdomen. 8. Mayflies are delicate-looking insects with 1 or 2 pairs of membranous, triangular wings, which are extensively covered with veins. At rest, the wings are held upright, like those of a butterfly. The hind wings are much smaller than the forewings and may be vestigial or absent. The second segment of the thorax, which bears the forewings, is enlarged to hold the main flight muscles. Adults have short, flexible antennae, large compound eyes, 3 ocelli and non-functional mouthparts. In most species, the males' eyes are large and the front legs unusually long, for use in locating and grasping females during the mid-air mating. In the males of some families, there are 2 large cylindrical, turban, eyes also known as turbinate or turbinate eyes, that face upwards in addition to the lateral eyes. 9. They are capable of detecting ultraviolet light and are thought to be used during courtship to detect females flying above them. 10. In some species all the legs are functionless, apart from the front pair in males. The abdomen is long and roughly cylindrical, with 10 segments and 2 or 3 long cerci, tail-like appendages, at the tip. Like Archeognaa, Archeognaa and Zygentoma, the spiracles on the abdomen don't have closing muscles. 11. 12. Uniquely among insects, mayflies possess paired genitalia, with the male having 2 ADD, penis-like organs, and the female 2 gonopores, sexual openings. 1. 4. Biology. Reproduction and life cycle. Mayflies are hemimetabolous, they have incomplete metamorphosis. They are unique among insects in that they molt one more time after acquiring functional wings. 13. This last but one-winged alate instar usually lives a very short time and is known as a subimago, or to fly fisherman as a dun. Mayflies at the subimago stage are a favorite food of many fish, and many fishing flies are modeled to resemble them. The subimago stage does not survive for long, rarely for more than 24 hours. In some species, it may last for just a few minutes, while the mayflies in the family Palingoniidae have sexually mature subimagos and no true adult form at all. 1. Often, all the individuals in a population mature at once, a hatch, and for a day or two in the spring or autumn. Mayflies are extremely abundant, dancing around each other in large groups, or resting on every available surface. 4. In many species the emergence is synchronized with dawn or dusk, and light intensity seems to be an important cue for emergence, but other factors may also be involved. Baetis intercalaris, for example, usually emerges just after sunset in July and August, but in one year, a large hatch was observed at midday in June. The soft-bodied subimagos are very attractive to predators. Synchronous emergence is probably an adaptive strategy that reduces the individual's risk of being eaten. 14. The lifespan of an adult mayfly is very short, varying with the species. The primary function of the adult is reproduction. Adults do not feed and have only vestigial mouthparts, while their digestive systems are filled with air. 13. Dillania americana has the shortest adult lifespan of any mayfly. The adult females of the species live for less than 5 minutes. 15. Mayflies, known locally as shadflies, swarm briefly in enormous numbers in Ontario. Male adults may patrol individually, but most congregate in swarms a few meters above water with clear open sky above it, and perform a nuptial or courtship dance. Each insect has a characteristic up and down pattern of movement. Strong wingbeats propel it upwards and forwards with the tail sloping down. When it stops moving its wings, it falls passively with the abdomen tilted upwards. Females fly into these swarms, and mating takes place in the air. A rising male clasps the thorax of a female. Each insect has a characteristic up and down pattern of movement. Strong wingbeats propel it upwards and forwards with the tail sloping down. When it stops moving its wings, it falls passively with the abdomen tilted upwards. Females fly into these swarms, and mating takes place in the air. A rising male clasps the thorax of a female from below using his front legs bent upwards, and inseminates her. Copulation may last just a few seconds, but occasionally a pair remains in tandem and flutters to the ground. 16. Males may spend the night in vegetation and return to their dance the following day. Although they do not feed, some briefly touch the surface to drink a little water before flying off. 16. Females typically lay between 400 and 3,000 eggs. The eggs are often dropped onto the surface of the water. Sometimes the female deposits them by dipping the tip of her abdomen into the water during flight, releasing a small batch of eggs each time, or deposits them in bulk while standing next to the water. In a few species, the female submerges and places the eggs among plants or in crevices underwater, but in general, they sink to the bottom. The incubation time is variable, depending at least in part on temperature, and may be anything from a few days to nearly a year. Eggs can go into a quiet dormant phase or diapause. 17. The larval growth rate is also temperature dependent, as is the number of molts. At anywhere between 10 and 50, these postembryonic molts are more numerous in mayflies than in most other insect orders. The nymphal stage of mayflies may last from several months to several years, depending on species and environmental conditions. 5. Around half of all mayfly species whose reproductive biology has been described are parthenogenetic, able to asexually reproduce, including both partially and exclusively parthenogenetic populations and species. 18. Many species breed in moving water, where there is a tendency for the eggs and nymphs to get washed downstream. To counteract this, females may fly upriver before depositing their eggs. For example, the female Tissa mayfly, the largest European species with a length of 12 centimeters, 4.7 in, flies up to 3 kilometers, 2 miles upstream before depositing eggs on the water surface. These sink to the bottom and hatch after 45 days, the nymphs burrowing their way into the sediment where they spend 2 or 3 years before hatching into subimagos. 19. When ready to emerge, several different strategies are used. In some species, the transformation of the nymph occurs underwater and the subimago swims to the surface and launches itself into the air. 4. In other species, the nymph rises to the surface, bursts out of its skin, remains quiescent for a minute or two resting on the exubi, cast skin, and then flies upwards, and in some, the nymph climbs out of the water before transforming. 20. Ecology. Rainbow trout are among the main predators of mayflies. Nymphs live primarily in streams under rocks, in decaying vegetation or in sediments. Few species live in lakes, but they are among the most prolific. For example, the emergence of one species of Hexagonea was recorded on Doppler weather radar by the shoreline of Lake Erie in 2003. 2.1. In the nymphs of most mayfly species, the paddle-like gills do not function as respiratory surfaces because sufficient oxygen is absorbed through the integument, instead serving to create a respiratory current. However, in low-oxygen environments such as the mud at the bottom of ponds in which Ephemera vulgata burrows, the filamentous gills act as true accessory respiratory organs and are used in gaseous exchange. 22. In most species, the nymphs or herbivores are detritivores, feeding on algae, diatoms or detritus, but in a few species, they are predators of chironomid and other small insect larvae and nymphs. 23-24 Nymphs of Pobola burrow into submerged wood and can be a problem for boat owners in Asia. 25 Some are able to shift from one feeding group to another as they grow, thus enabling them to utilize a variety of food resources. They process a great quantity of organic matter as nymphs and transfer a lot of phosphates and nitrates to terrestrial environments when they emerge from the water, thus helping to remove pollutants from aqueous systems. 5 Along with caddisfly larvae and gastropod mollusks, the grazing of mayfly nymphs has a significant impact on the primary producers, the plants and algae, on the bed of streams and rivers. 26 The nymphs are eaten by a wide range of predators and form an important part of the aquatic food chain. Fish are among the main predators, picking nymphs off the bottom or ingesting them in the water column, and feeding on emerging nymphs and adults on the water surface. Carnivorous stonefly, caddisfly, alderfly and dragonfly larvae feed on bottom-dwelling mayfly nymphs, as do aquatic beetles, leeches, crayfish and amphibians. 27 Besides the direct mortality caused by these predators, the behavior of their potential prey is also affected, with the nymph's growth rate being slowed by the need to hide rather than feed. 26 The nymphs are highly susceptible to pollution and can be useful in the biomonitoring of water bodies. 4 Once they have emerged, large numbers are preyed on by birds, bats and by other insects, such as Rhamphomya longicauda. 5 Mayfly nymphs may serve as hosts for parasites such as nematodes and trematodes. Some of these affect the nymph's behavior in such a way that they become more likely to be predated. 28-29 Other nematodes turn adult male mayflies into quasi-females which haunt the edges of streams, enabling the parasites to break their way out into the aqueous environment they need to complete their life cycles. 30 The nymphs can also serve as intermediate hosts for the horsehair worm Paragordius varius, which causes its definitive host, a grasshopper, to jump into water and drown. 31 Effects on ecosystem functioning. Mayflies are involved in both primary production and bioturbation. A study in laboratory simulated streams revealed that the mayfly genus Centroptilum increased the export of paraphyton, 32 thus indirectly affecting primary production positively, which is an essential process for ecosystems. The mayfly can also reallocate and alter the nutrient availability in aquatic habitats through the process of bioturbation. By burrowing in the bottom of lakes and redistributing nutrients, mayflies indirectly regulate phytoplankton and epibenthic primary production. 33 Once burrowing to the bottom of the lake, mayfly nymphs begin to billow their respiratory gills. This motion creates current that carries food particles through the burrow and allows the nymph to filter feed. Other mayfly nymphs possess elaborate filter feeding mechanisms like that of the genus Isonychia. The nymph have forelegs that contain long bristle-like structures that have 2 rows of hairs. Interlocking hairs form the filter by which the insect traps food particles. The action of filter feeding has a small impact on water purification but an even larger impact on the convergence of small particulate matter into matter of a more complex form that goes on to benefit consumers later in the food chain. 34. Distribution. Mayflies are distributed all over the world in clean freshwater habitats, 35 though absent from Antarctica. 36 They tend to be absent from oceanic islands or represented by 1 or 2 species that have dispersed from nearby mainland. Female mayflies may be dispersed by wind, and eggs may be transferred by adhesion to the legs of waterbirds. 37 The greatest generic diversity is found in the neotropical realm, while the Holarctic has a smaller number of genera but a high degree of speciation. Some 13 families are restricted to a single bioregion. 38 The main families have some general habitat preferences. The Batidae favor warm water, the Heptagenidae live under stones and prefer fast-flowing water, and the relatively large Ephemeroptera make burrows in sandy lake or river beds. 35. Conservation. The nymph is the dominant life history stage of the mayfly. Different insect species vary in their tolerance to water pollution, but in general, the larval stages of mayflies, stoneflies Plecoptera, and caddisflies Trichoptera, are susceptible to a number of pollutants including sewage, pesticides and industrial effluent. In general, mayflies are particularly sensitive to acidification, but tolerances vary, and certain species are exceptionally tolerant to heavy metal contamination and to low pH levels. Ephemeroptera are among the most tolerant groups and Cyclonuridae and Canidae the least. The adverse effects on the insects of pollution may be either lethal or sublethal, in the latter case resulting in altered enzyme function, poor growth, changed behavior or lack of reproductive success. As important parts of the food chain, pollution can cause knock-on effects to other organisms. A dearth of herbivorous nymphs can cause overgrowth of algae, and a scarcity of predaceous nymphs can result in an overabundance of their prey species. 39 Fish that feed on mayfly nymphs that have bioaccumulated heavy metals are themselves at risk. 40 Adult female mayflies find water by detecting the polarization of reflected light. They are easily fooled by other polished surfaces which can act as traps for swarming mayflies. 10. The threat to mayflies applies also to their eggs. Modest levels of pollution in rivers in England are sufficient to kill 80% of mayfly eggs, which are as vulnerable to pollutants as other life cycle stages. Numbers of the blue-winged olive mayfly betas have fallen dramatically, almost to none in some rivers. The major pollutants thought to be responsible are fine sediment and phosphate from agriculture and sewage. 41. The status of many species of mayflies is unknown because they are known from only the original collection data. 4 North American species are believed to be extinct. Among these, Pentagonea robusta was originally collected from the Ohio River near Cincinnati, but this species has not been seen since its original collection in the 1800s. Ephemera compar is known from a single specimen, collected from the foothills of Colorado, in 1873, but despite intensive surveys of the Colorado mayflies reported in 1984, it has not been rediscovered. 42. The International Union for Conservation of Nature (IUCN) Red List of threatened species includes 1 mayfly, Tasmanophlebia lacuscorule, the large blue lake mayfly, which is a native of Australia and is listed as endangered because its alpine habitat is vulnerable to climate change. 43. Taxonomy and phylogeny. Fossil adult Mycoladia longimanus Coxoplectoptera, Mycolatiidae, from the Lower Cretaceous Crotto Formation of Brazil, circa 108 Maya. Ephemeroptera was defined by Alpheus Hyatt and Jenny Maria Arms Sheldon in 1890-1. 44-45 The taxonomy of the Ephemeroptera was reworked by George F. Edmonds and J. R. Traver, starting in 1954. 46-47 Traver contributed to the 1935 work The Biology of Mayflies, 48, and has been called the first Ephemeroptera specialist in North America. 49. As of 2012, over 3,000 species of mayfly in 42 families and over 400 genera are known worldwide, 50-51 including about 630 species in North America. 52 Mayflies are an ancient group of winged pterygote insects. Putative fossil stem group representatives, e.g. Syntenopteroidea like Lithonura lamirae, are already known from the late Carboniferous. 53 The name Ephemeroptera is from the Greek ephemeros, short-lived, literally, lasting a day, cf. English, ephemeral, and pteron, wing, referring to the brief lifespan of adults. The English common name is for the insect's emergence in or around the month of May in the UK. 54 The name shadfly is from the Atlantic fish the shad, which runs up American East Coast rivers at the same time as many mayflies emerge. 55-56 From the Permian, numerous stem group representatives of mayflies are known, which are often lumped into a separate taxon Promoplectoptera, e.g. including Proterisma permianum in the Proterismatidae, 53, and Mysthodontidae. The larva of Promoplectoptera still had 9 pairs of abdominal gills, and the adults still had long hindwings. Maybe the fossil family Crederismatidae from the Lower Cretaceous Crotto Formation of Brazil also belongs as the last offshoot to Promoplectoptera. The Crotto outcrops otherwise yielded fossil specimens of modern mayfly families are the extinct but modern family Hexagenidae. However, from the same locality the strange larvae and adults of the extinct family Mycolatiidae, order Coxoplectoptera, have been described. 57 which represents the fossil sister group of modern mayflies, even though they had very peculiar adaptations such as raptorial forelegs. The oldest mayfly inclusion in amber is Cretinetta gerashini, Leptophlebeidae, from the Lower Cretaceous of Siberia. In the much younger Baltic amber numerous inclusions of several modern families of mayflies have been found: Ephemeridae, Podomanthidae, Leptophlebeidae, Ametropodidae, Cyclonuridae, Isonichidae, Heptagenidae, and Ephemeralidae. 58 The modern genus Neoephemera is represented in the fossil record by the Epresian, 59, species N. antiqua from Washington State. 60. Grimaldi and Engel, reviewing the phylogeny in 2005, commented that many cladistic studies had been made with no stability in Ephemeroptera suborders and infraorders. The traditional division into Schistosnota and Panota was wrong because Panota is derived from the Schistosnota. 53 The phylogeny of the Ephemeroptera was first studied using molecular analysis by Ogden and Whiting in 2005. They recovered the Betidae as sister to the other clades. 61 Mayfly phylogeny was further studied using morphological and molecular analyses by Ogden and others in 2009. They found that the Asian genus Cyphloriscus was sister to all other mayflies. Some existing lineages such as Ephemeroidea, and families such as Amelatopsidae, were found not to be monophyletic, through convergence among nymphal features. 62. The following traditional classification, with 2 suborders Panota and Schistosnota, was introduced in 1979 by W. P. McCafferty and George F. Edmonds. 63. The list is based on Peters and Campbell, 1991, in Insects of Australia. In art. The Dutch Golden Age author Augurijus Clusius Aukert Kloot illustrated some mayflies in his 1634 De Hemerobio, on the mayfly, the earliest book written on the group. Maerten de Vos similarly illustrated a mayfly in his 1587 depiction of the 5th day of creation, amongst an assortment of fish and water birds. 65-66. In 1495 Albrecht Dürer included a mayfly in his engraving The Holy Family with the mayfly. 67 The critics Larry Silver and Pamela H. Smith argue that the image provides an explicit link between heaven and earth, to suggest a cosmic resonance between sacred and profane, celestial and terrestrial, macrocosm and microcosm. 68. In human culture. Mayfly in art. Mayflies drawn. By Augurijus Clusius, in De Hemerobio, 1634. Mayfly by Jan Saudeller after Maerten de Vos, detail from the 5th day, the creation of the birds and fishes, circa 1587. Albrecht. Dürer's. Engraving the Holy Family. With the Mayfly, 1495. Detail of Mayfly, in lower right corner of Albrecht Dürer's engraving The Holy Family with the Mayfly, 1495. Mayflies in Sunset Dance, by Philip Henry Goss. In a Victorian edition of Gilbert White's Natural History of Selborne. In literature. The ancient Greek biologist and philosopher Aristotle wrote in his History of Animals that Bloodless and many-footed animals, whether furnished with wings or feet, move with more than 4 points of motion, as, for instance, the dayfly ephemeron moves with 4 feet and 4 wings. And, I may observe in passing, this creature is exceptional not only in regard to the duration of its existence, whence it receives its name, but also because though a quadruped it has wings also. 69b. The ancient Roman encyclopedist Pliny the Elder described the